And so we move on to Joshua McGrath. He will talk about immunoglobulin codon optimality, which is a very interesting topic. I really liked his paper when it came out. He's a postdoc currently in the Patrick Wilson lab at Weill Cornell Medicine in New York, where he's working on a Banting postdoc fellowship, and he's broadly interested in antibody responses and antigen-specific B-cell repertoires in all contexts, infection, vaccination, and autoimmunity. Looking forward to your talk.
Sorry, I didn't realize I was muted. Can everybody see my screen okay?
Yes, and we can hear you.
Great, thank you. I just want to say thank you so much for the invitation to give this talk today. I'm really excited to share some of our recent work where we investigated some evolutionary relationships between mutability, somatic hypermutation, and immunoglobulin codon optimality. This was recently published in Molecular Cell. So this will be no news to this crowd, but I'll briefly go through it anyways. Immunoglobulins, BCRs, antibodies, in their secreted form, mediate various functions in health and disease, neutralization, ADCC, etc. And in order to perform these functions, they typically must be secreted at a high volume per cell in order to accumulate at enough concentration to mediate those functions. And the functions that antibodies perform are largely dependent on the specificity of their variable regions. As we all know, heavy and light chain come together to form a paratope, which can bind to epitopes uh, and facilitate those various interactions that I described previously. And the diversity within those variable regions is generated through three main processes: VDJ recombination, junctional diversification, and somatic hypermutation of both heavy and light chain genes. And ultimately, this can produce a theoretical 10 to the 15 unique germline BCR configurations with approximately 10 to the 8 to 10 to the 10 unique B cell variants circulating in any individual at any given time, which is staggering to say the least. So we have these two aspects. On one hand, we have this requirement for various forms of diversity, which are encoded by the DNA: rhombinal germline diversity, junctional diversity, hypermutative diversity. We also have this requirement for extensive antibody secretion on a per cell basis. And this can also be encoded at the DNA level through codon optimality. And in the lab, we often make recombinant antibodies. We modify their sequences, and this led us to ask the question: when we modify these sequences, are we changing the codon optimality? And more fundamentally, how has variable region diversity co-evolved in parallel with codon optimality, which are both DNA encoded features? And does this affect protein expression efficiency? And so with this, we had three hypotheses. The first is that germline VDJ genes exhibit variation in codon optimality, very simply. The second, that somatic hypermutation can modify that optimality. We hypothesized initially that this would be to their detriment. And finally, that VDJ codon optimality can influence antibody expression efficiency in vivo and in vitro. Okay, so how do we measure codon optimality? This was a challenge that we had at the beginning. Typically and canonically, the way that it's measured is through usage bias in defining codons that are more optimal as those that are used more frequently. And in the 1980s, Sharp and Lee came up with this metric, the codon adaptation index. And essentially what this measures is codons that are used within a particular coding sequence versus the frequency at which they are used across all coding sequences within a given species. So essentially what it does is for a given amino acid sequence, it looks at the codons that are used. It looks at the frequency of the codons that are used divided by the most frequently used codons, and it comes up with a codon adaptation index value between 0 and 1, which represents how synonymously optimized that coding sequence is. But notably, if you have two disparate proteins, A and B, they have completely distinct amino acid sequences, but they both use their most optimal or most frequently used set of codons. They will both have codon adaptation indices of 1, which in our view at the beginning of this project seemed to minimize some of the diversity that we might see. And so we kept looking in the literature to see whether there was other alternatives. And more recently, there's been a number of studies, a few of them shown at the bottom, one highlighted, that define codon optimality as a relationship to mRNA longevity. And they define optimal codons as those which contribute positively to mRNA stability. And to just take you through one of the papers that was very important to our study, published in 2019, the authors took various cell types, human cell types, they treated them with a transcriptional inhibitor, and then they, over time, monitored mRNA decay. And then they measured the frequency of occurrence of individual codons within the measured mRNAs, and they correlated the mRNA stability against the occurrence of those codons. And they found that some codons had a positive correlation with stability and some had a negative correlation. And they were able to do this across various model systems that we can see at the top right, various human cell types, various experimental systems, and they were able to define what are known as codon stability coefficients, which is the Pearson correlation between mRNA stability and the occurrence of a particular codon. And across all of these experimental systems, there was very relatively consistent results, suggesting that you can define a contribution of a particular codon to mRNA longevity. And this is done in multiple papers for human cells. It's been done also in various model systems, Xenopus, et cetera. And so we decided to move forward using this system because it helped maintain the diversity that seemed to be lost in the codon adaptation indices. And so this is the basic method that we used. This is for germline genes, but the same premise applies to when we look at BCR repertoires. We took spliced human germline immunoglobulin coding sequences from IMGT. We broke them down into their individual codons. And then for our primary readout, we took the average of these 6 assays from Wu et al. And we assigned each codon its CSC value. And then we took the average of those values over the entire length to generate one single CSC average value for each sequence. And, we did this for not only this paper, but using values from other papers as well, using codon adaptation indices. But I'm just going to focus on the CSC averages from this paper because that was the primary readout from our study, because they kind of analyzed these CSC values in the most robust fashion. Okay, so what do we see in terms of germline sequences? On the left-hand side, we can see alleles. So we've got V alleles in yellow, D and J alleles in green and blue, respectively, and then constant gene alleles, constant region alleles in pink. And there is diversity. With sequence diversity, you would expect diversity in codon optimality. So there's no surprise there. When we started binning the V genes, and we focused on the V genes for a lot of this because they comprise the bulk of the VDJ sequences in terms of length. When we binned the alleles in terms of V gene, we found that there was definitely some bias in terms of the segregation of different families. On the right-hand side, we can see that IGHV4 genes tended to have a very high optimality. Excuse me. V3 genes had a low optimality, and 1s and 2s came somewhere in between. And when we looked at these alleles, when we looked at not only heavy chain, but light chain, as well as T cell receptor genes, we found that in particular for IGHVs and IGHJs, these were highly optimal. And this is by comparing these values to all human indexed ORFs from the CCDS database, finding that IGHVs were within the top 25% and IGHJs within the top 2% of indexed human ORFs, which was really exciting. One other thing to note is that when we frame shifted these IGHVs by one nucleotide, disrupting that natural open reading frame, we saw a dramatic reduction in optimality, kind of supporting the use of this optimality metric, requiring something that is evolved rather than disrupted. Okay, so the next thing that we did was we wanted to understand, okay, what's the difference between functional genes and pseudogenes? With the hypothesis that if we are measuring optimality, pseudogenes in humans, by virtue of not being under functionality associated selective pressures, should have a lower optimality because you are getting some, effects of time and random mutation accumulating within those sequences. And that's indeed what we found when we looked at heavy chain and light chain V gene sequences. There was a significant reduction in the optimality of pseudogenes as compared to functional genes in all cases, suggesting that these V genes maintain codon optimality under functionality-driven selective pressures. Okay, so this is great. What about the relevance? Does this have any consequence in vivo. And this was something that was tricky to figure out how to assess. But what we did in the end was we collaborated with Dr. George Georgiou at the University of Texas at Austin and Dr. Jian Park, who had this really unique and great protocol, IG-seq. And what they did was they had a data set where they took serum from vaccinated donors, influenza vaccinated donors. They purified out the hemagglutinin and hemagglutinin specific and non-specific IgG fractions, and they fragmented them for mass spec. They also, from the same samples, performed IgH sequencing, and by pairing these two data sets together, they were able to identify IgG clones within the serum Ig repertoire. And when we averaged those for individual VH genes, and we correlated that against the germline optimality data for those individual VH genes, we found that in both the IgG specific or the H3 specific and non-specific fractions, there was a positive relationship, suggesting that IgHV codon optimality may subtly influence global serum IgG VH usage. Of course, there are lots of other factors that go into the effect on individual clonotypes, such as whether they're contributing to a particular disease that is circulating within the individual at that given time. But it looks like there is a subtle effect on the global level, which was very exciting to see. Okay, so another feature that we wanted to look at is mutability. So in the future, our ultimate goal was to look at the effect of somatic hypermutation, but somatic hypermutation, uh, the predilection to undergo mutation is encoded at the DNA level through the presence of hotspots and cold spots within CDRs and framework regions, as we're all aware. And so we wanted to understand whether this mutability correlates with optimality. And so to assess this, we used the Shazam package from the Kleinstein group. In particular, we looked at the mutability function. So we took every 5-mer within our sequences, and we looked at the mutability of the central nucleotide and then took an average over the length of each sequence to get our mutability score average, which we correlated against our optimality score average on the x-axis. I'm sorry, that title is missing. The x-axis is optimality score. What we found is that in heavy chain genes and in light chain genes, there was an inverse relationship between mutability and optimality, suggesting that these two features are inversely related. And so, okay, we wanted to dig into this a little bit further. When we break these sequences down into CDRs and framework regions, we know that CDRs are typically more mutable than framework regions. And so with this common understanding, we would expect that their optimality would be lower based on this established relationship. And that is, in general, what we saw. The CDRs in pink tended to have a lower optimality than the framework regions in gray. So that further supports that inverse relationship we see across the whole sequence. What happens if we play with these systems and we artificially maximize or minimize mutability? What happens then? So what we did was we took, if we focus on the left-hand side, 6 V genes. We took their original sequences. The 3 on the left have high initial optimality. The 3 on the right have low initial optimality. And then we did 2 things. We neutralized mutability by running these sequences through a codon optimizer, essentially replacing the codon usage with those that are reflective of the overall codon usage within human genes. So these are no longer specified for mutability. And on the other hand, we actually increase the number of hotspots in either the CDRs alone or in framework regions as well. So we're increasing mutability to the right and we're decreasing mutability to the left, or neutralizing mutability. And to sum it up, if you look on the right-hand side, overall, these two values tended to move in opposition with each other. When you increase mutability, optimality went down quite significantly, suggesting that this artificial scaling supports the natural inverse relationship between mutability and optimality that we see within germline genes. And we thought this was pretty exciting. Okay, so we've looked at germline features up until this point, and the next goal was to look within BCR repertoires, somatic hypermutation. But one interesting opportunity we had in between was to simulate somatic hypermutation using ImmuneSim from the Greif and Reddy labs. And so what we did was we ran the simulations at various somatic hypermutation probabilities for heavy chains and light chain genes. And we correlated the number of somatic or simulated VDJ mutations against optimality. We found that in all 3 cases, there was a linear attenuation of optimality with increasing mutation. Okay, so this is in silico. What happens in vivo? And it turns out we do see that when we look at the heavy chain genes. This is an in-house data set. We did 5' 10x genomic sequencing on a tonsil sample from a 16-year-old female donor, and we quantified our VDJ mutations within the heavy chain, finding that as you increase the number of VDJ mutations, the optimality tends to decrease by virtue of a significant negative correlation. When we bin the sequences according to mutational load, we see the same thing. In purple, as you increase the number of mutations, the optimality goes down. But importantly, when we revert them back to germline, there's no relationship. And this suggests that effect is due to mutation and not due to selection of sequences in highly mutated clones that have differing optimalities. And on the right-hand side, when we revert them back to germline, you see a significant increase in optimality. Interestingly, though, we didn't see any difference for the kappa and lambda chain sequences, which stumped us for a long time. But I think what is a good explanation on the next slide. Going back to heavy chains, so that was one in-house data set. We corroborated this with another in-house data set looking at lung-draining lymph node total B cells, as well as within bone marrow plasma cells from a published paper from Dr. Ali Elabidi's group. Again, with increasing mutational load, we see de-optimization that is fully germline reversible. And this is not just in humans. So when we infected mice with influenza, we looked at their heavy chain genes after 32 weeks. We found that as you increase mutation, again, in the heavy chain, we see de-optimization. We also looked at zebrafish from a published article back from 2011 from Dr. Stephen Quake's group. And in this case, we changed our protocol just a little bit, but we were looking at CDR3 optimality binned by framework 3 mutational burden. But again, we see the same thing. As you increase mutation, you get reduced optimality. So overall, across various human tissues and various species, we are seeing a pretty consistent effect within the heavy chains. As you increase mutation, you see reduced optimality. So why not light chain? So this was something that was mentioned in every presentation I gave. This was something that was brought up during the review process. So it became a priority to understand. And we hypothesized that it was due to the different germline optimalities of these chains. So if you just focus on the gray bars, you can see that the heavy chain germline optimality mean is much higher than the kappa chain or the lambda. And we hypothesized that this had something to do with it. So what we did was we just filtered to sequences that had a common germline optimality across all chains. So within the yellow band, just the heavy kappa and lambda sequences within the yellow band that were germline. And we looked at how they de-optimized in response to mutation. And it turns out that they all de-optimized in exactly the same rate, regardless of chain. And what this suggests is that initial optimality is pretty key to how they de-optimize in response to the same number of mutations. And so reciprocally, we wanted to ask if we normalize the mutational load to just those that had 31 to 40 mutations, what do we see then? Well, what we see is that those that have on the right-hand side of the graph a higher germline optimality undergo a greater de-optimization than those that have a lower germline optimality. And so overall, this really suggests that the germline optimality is critical in understanding why heavy chains de-optimize as compared to light chains, which do not seem to de-optimize. Okay, so we've been looking at the global repertoires so far. What about within clonal families? And this paper from Dr. Elabidi's group had a very extensive collection of and delineation of spike-positive clonal families from SARS-CoV-2 mRNA vaccinated donors. This gave us a really good opportunity to assess this. And in the overall repertoires, we're seeing this negative correlation. And if you look in the middle here, we can see that there are a number of clonal families, which this is not all of them, it's just a select few, that de-optimize in response to somatic hypermutation, as we see in the overall repertoires. On the right-hand side, we can see that some families actually seem to increase in codon optimality. But on balance, when we look at all the clonal families in the volcano plot at left, the vast majority of them tend to de-optimize in line with what we see on the whole repertoire level, suggesting that mutational de-optimization is observable within large heavy chain clonal families. This is a rather large SARS-2 data set. We also showed this for HIV-1 specific heavy chain clonal families, although that data is not shown here. We next wanted to ask how the biased nature of mutational targeting could relate to de-optimization. So we know that CDRs have a greater predilection to undergo somatic hypermutation than framework regions by virtue of their antigen-facing position. Um, and so we wanted to ask whether the differential targeting of CDRs versus framework region had any effect on the de-optimization process. So what we did firstly was we took our original sequences from various tissues, and these have mutational distributions that are natural. We then reverted them to germline, and then we reintroduced within those same germline sequences the same number of mutations, but in a totally random pattern. We did this 10 times per sequence, and we did this for almost 800,000 sequences across various data sets that we analyzed in the paper: tonsils, lung draining lymph nodes, bone marrow, and other lymph nodes from published data. And what we found was that if you look in the middle, you can see that in the purple, that's our original de-optimization rate. When we germline revert, we lose that relationship altogether. And then when we reintroduce random mutations, it's a greater de-optimization, which we thought was really interesting. And so in thinking this through, if you randomly remutate, you're removing the bias. And in this case, removing the bias would theoretically mean increasing a greater mutational frequency within the framework regions. So is that the case? Does targeting mutations within the framework regions have a greater de-optimizing effect than CDRs? And so to do this, we took our tonsil VDJ sequences, we looked at the codons that were being targeted by mutation, and we looked at what they were initially, what the germline version of the codon and the mutated version of the codon, and we looked at the optimality of that transition. So as they undergo mutation, do those codons change from high to low optimality, low to high optimality, et cetera, binned by framework region and CDR? And what we found in the middle is if we look at framework regions as a whole, they started with very high optimality, and when they mutated, there was a dramatic reduction. And by comparison, CDRs, which had a low initial optimality, increased by virtue of somatic hypermutation. And we thought this was really interesting. When you break it down, for CDRs, there's a little bit of a difference between the individual CDRs, with CDR2 having a very low initial optimality, CDR1 being relatively neutral and not changing very much. But overall, the effect is very distinct from framework regions, which all have a high initial optimality and all undergo a great amount of de-optimization in response to somatic hypermutation. Overall, suggesting that the mutation of framework regions has a much greater de-optimizing effect than CDRs. Which makes sense evolutionarily. If you're targeting most of your mutations to CDRs, this will have less of an effect on codon optimality than would mutating framework regions, which won't have as much of a benefit on specificity or effector function anyways. And the other thing to notice here is this also provides some really interesting data about our inverse relationship between mutability and optimality. If you just look at the germline on the left-hand side there, highlighted by the arrow, CDR2 and CDR1 optimalities, and you compare that to the overall optimalities of the CDR1 and CDR2 within those regions, you can see that in both cases, they are quite a bit lower. And overall, by virtue of the fact that these codons were mutated, they are more mutable than their neighbors, which are not mutated. And again, this is over hundreds of thousands of cells, so it's a general trend, suggesting that the cells that are mutated and therefore inherently more mutable have a lower optimality than their less mutable neighbors, supporting that earlier inverse relationship. Finally, earlier we looked at in vivo correlates between optimality and antibody expression. We also wanted to look in vitro. Now that we have these BCR data sets, we can express some monoclonal antibodies. So we did two sets. One was from lung draining lymph nodes. One was from the blood of SARS-2 infected individuals, overall numbering 15 antibodies. We made the original versions of the monoclonals, and then we made synonymously de-optimized, so same amino acid sequence, but the heavy and light chain plasmids were de-optimized to a maximum extent. And ultimately, what we found was that on both the mRNA and protein levels, there was reduced expression of the monoclonal in the synonymously de-optimized form, suggesting that this optimality metric can give us some indication of expression efficiency. So overall, what we were able to show in this paper is that germline VDJ genes do exhibit variation in codon optimality, with IGHVs and IGHJs being quite optimal as compared to, for instance, T cell receptor genes, which had comparatively lower optimality, and they rank quite highly among all indexed human ORFs. Uh, we also showed that optimality appears to be maintained by functionality. And mutability is inversely related to optimality, with optimality being lower in CDRs than in framework regions, and in particular lower in uh, for mutation or for codons that are actually targeted by somatic hypermutation than in the surrounding regions. We found that somatic hypermutation de-optimizes heavy chain VDJs in humans and non-humans, various human model or various human data sets and non-human models. And this is primarily due to their higher germline optimality than light chains. We found that there was a bias, that the bias targeting to CDRs can help partially mitigate de-optimization, and that synonymous de-optimization could reduce monoclonal expression in vitro, and that optimality may subtly influence expression in vivo. And overall, our takeaway from this is that immunoglobulin genes have evolved to support diversity and affinity maturation, despite some resulting variation in codon optimality that may subtly affect expression levels in vivo. With that, I'd like to say thank you again for inviting me to give this talk. I'd like to thank Patrick Wilson, my PI, who gave me a lot of freedom on this project. Our bioinformaticians, Chloe and Jordan, who I could not have done this paper without. And Dr. Giorgio and Garami at Weill Cornell, who helped provide the tonsils. Doctors Madriaga and Kent at the University of Chicago for providing the lung draining lymph nodes. And of course, Doctors Giorgio and Park from University of Texas at Austin for the IG-seq data and all of our sponsors. And with that, I would like to take any questions. Thank you so much.
Thanks, Joshua. Extremely exciting talk, and I don't want to know how much work this was. A very quick question because I need to leave in 4 minutes, but
Okay.
and I have so many questions. So, would it be also an idea, because so you correlated your data with George's IG-seq technology, uh,
Mhm.
which I think has problems in terms of sampling. So the correlation might also be due to, you have a lack of what it can actually see because it's reference
Sure.
based and so on. Did you correlate there, by the way, with different isotypes, or this was isotype unspecific, the correlation there?
So the correlation with George's data was only IgG because that was what they were focusing on for their data set. And we were using it as like an opportunistic collaboration.
Yeah.
What I will say in terms of isotype was that what I didn't show, it's in the supplement of the paper, was we looked at when we binned cells by isotype,
Hmm.
we saw that, for instance, IgA and IgG1 had a
Hmm.
slightly lower optimality than IgM and IgD.
Hmm.
But this was pretty suspect because those are class switch cells, and class switch cells would have an increased predilection to undergo mutation. So when we removed all the mutated cells, we lost that relationship. And overall, what we were able to conclude was that, at least on the cellular level, there didn't appear to be differences in terms of what cells were selected or what clones were selected for isotype switching when you get rid of that mutational burden.
Hmm. 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 Very interesting. Would it be also an idea because there was some French labs who published a few years ago microfluidics technology to look at secretory rate of plasma cells, for example. Would that be an idea to correlate that maybe so the secretion rate, really to correlate that could be, I mean, in the future if one were to continue that
I
correct?
mean, yeah, that's a great idea. I was not aware of that technology. There was it was a bit of a struggle to figure out what the best way to look at
Yeah.
in vivo metrics was.
Yeah.
And that's not something I was aware of. So we'll definitely look into that.
Yeah, yeah. And so the last slide that you had with the expression that actually I had this question before and then you almost gave the answer. So could one say codon optimality could be another correlate for antibody developability, maybe also what the therapeutic fields is for after? Question mark.
Yeah, absolutely. I definitely think that in terms of developability, people are already looking at codon optimality, and
Okay.
what we're looking at here is a different metric of optimality. One
Yeah.
thing that we, haven't done is do a head-to-head comparison of the how well our metric correlates against CAIs, how well it correlates some of the other metrics that we came up here
Yes.
in terms of predicting
Yes.
expression efficiency. So,
That,
and the other thing is these are completely synonymously de-optimized.
mm,
And so certainly the difference in optimality between the original and the de-optimized in this scenario will be typically much greater than any two natural antibodies, which both are using, a relatively optimal schema of codons. So I definitely think that optimality is an important role in that developability pipeline. One of the extensions that I really wanted to pursue at one point was looking at single chain Fabs
yes.
or SCFVs, which the a greater proportion of those constructs are VDJ, right?
Yeah.
Their antibodies are largely constant regions, so will largely be static. And to note, these are not de-optimized at the constant regions; these are only de-optimized at the VDJs. So when you de-optimize an entire VDJ of a single chain Fab, I would expect a even more drastic effect. Yeah, I agree. I think that developability, especially in terms of antibody fragments, optimality is an important consideration.
Super interesting. And then last question from me. At one of your first slides, when you looked at the optimality per V gene, I think it was. Um,
Mhm.
and I think you said it in the paper, but maybe you can remind me, or even in the slides. Was that correlated to also the frequency of? Yes. So no. And so is this ranking also consistent with how often we see these V genes used in the repertoire?
No, it is not. That is in the
Supplementary.
supplement of the paper. When we looked at IgD cells that were
Hmm.
not mutated,
Yeah.
so germline IgD cells,
Yeah.
we did not find that there was any enrichment of highly optimal versus lowly optimal V genes.
That is a bit interesting, maybe, no?
Oh, no, it's definitely interesting. I mean, we hypothesized that there would be,
Yeah, right. Yeah.
but certainly, I mean, especially within naive cells, you would expect there to be relatively unbiased. Within class switch cells, you would expect there to be a lot more going on that would
Yeah.
influence the usage rates. But within naive cells, you would think that there would be, where we saw a trend towards an increase, but not a significant increase. So, if there are other factors, we have potentially an increased usage of VH169 because
Yeah.
it's good at binding a lot of epitopes that are relevant to human infectious
Exactly.
disease, right?
Exactly.
So
Yeah. 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 Yeah.
surprising, but I agree.
Okay, interesting. Okay. Um, unless there are no burning questions, I would like to close here.
Okay,
Okay.
stop sharing.
Um, thanks so much again for this beautiful talk, and we will be back in September with another seminar. Thanks so much, Joshua. Thanks so much.
Thank you.
Thanks, everyone. Bye bye. Bye bye.
Bye bye.